Hello, this is Mrs. Murdock, and this is an AP Biology review session for the Unit 4 test on, oh, it hurt purple. For the Unit 4 test, which is on the cell cycle, which includes like mitosis and the S and G1 and G2 phases and all of that, and also on cell communication, which is also part of this unit. Cell communication, which includes things like how hormones tell cells what to do, and signal transduction pathways, and also the immune system. Okay, so these are questions that I pulled from your progress check on Unit 4 in the AP classroom. So I thought it would be good to go over. I didn't pick very many. I just picked a few because I thought these were really good examples of good graphics and good examples of questions where you really have to read it carefully and decode the language to be able to answer the question correctly. So this first one says researchers grew seedlings of corn in loose versus compact sand, right? So here's the loose sand is here, and here's the compact sand is here. And they measured the amount of time required for the cells in the growing root tips of the seedlings to double in number. So mean cell doubling time, right, in hours, cell doubling time, okay. The mean cell doubling times for the two groups of seedlings are shown as bars. All right, so now let's go over to the question. And the question says, based on the sample means, which of the following conclusions about the cells in the growing root tips of the Zea maïs seedlings is best supported by the results of the experiment? So basically, they want you to get the right sentence that actually goes with the data that they're showing, right? That would be a valid conclusion that matches the data. So it's really easy to fall into a trap here. You got to really know what it is you're looking at. So the first choice is the cells of the root tips grow to larger sizes when the seedlings are planted in compact sand than when the seedlings are planted in loose sand. Okay, so when I first did this, I'm going to admit that I thought, oh, that's right? Because doesn't that mean that it's in compact sand it's taking less time, right, for them to grow larger? Except you got to read it carefully, because look at what that choice says. It says the cells are growing larger sizes. Is that what they're measuring? No. They're measuring cell doubling time. How fast the cells are going through mitosis, essentially, right? Which is completely different than a cell growing larger. So that choice is out. Not correct. We're not measuring how big the cells are getting. We're measuring how fast they are doubling in quantity, okay, not in size. Okay, the double in number. The B says, the average rate of mitotic cell division is greater for the root tips growing in loose sand than for the root tips growing in compact sand. Okay, let's decode that. The rate for mitosis cell division is greater, faster in loose sand? I don't think so. It's taking more time, right? So that's not going faster if it's taking more time to double. It's taking, sorry, so the shorter amount of time means this is going faster, right? In compact sand, it's not taking as many hours for the cells to double than it is in the loose sand. It's taking more hours here. So this is slower and this is faster. Good? That's another thing you got to be careful of. It's an easy trap to fall into. Okay. So now let's go to the C. The average cell cycle time is greater for the root tips growing in compact sand than for the root tips growing in loose sand. Okay, so again, cell cycle time. Remember the cell cycle is going through all of those stages. Are we going through it faster? In other words, is the mitosis faster again? So is it faster in compact sand? Not necessarily, because we don't know which part of the cell cycle they're asking about, okay? More cells are produced per unit time in the root tips growing in compact sand than in the root tips growing in loose sand. Okay. So more cells are produced per unit of time in the root tips here. Yes, that's the one. That is the clearest one, right? Because what we're really looking at is mitosis here. We don't want the entire cell cycle being measured, just the mitosis, right? Okay, so that first question, D is the right answer. Yes. More cells produced per unit of time, which is a rate, right? Per unit of time, it's faster in the compact sand. And we've already marked up the graph to prove to ourselves that the cell doubling time is less. Therefore, it's going faster in the compact sand than it is in the loose. Okay, so that's what you have to do with these questions. I write all over the questions. I draw pictures for myself. I notate it for yourself so that it will help you figure it out. That's good advice there. Okay, let's look at the next question. All right. This is a very common graph in cell cycle type AP level questions. And what this graph is measuring is the amount of DNA per cell at the different stages here. And they're not telling you what those stages are. They're just saying that in this first stage, there is 1 times the amount of DNA. In the second stage, the amount of DNA doubles, right? It increases 2-fold. In that third stage, it just stays at that doubled amount. And in the fourth stage, something happens that cuts it back in half again. Okay, so again, remember the cell cycle. In this case, it's going to be more useful for us because we're talking about all of the cell cycle sections here. Okay, remember we have G1, we have S, we have G2, and we have M, where the cells divide. Okay, G1 is growth, right? What happens in S? The most important thing that happens in the S phase. DNA replication. Beautiful. DNA replication happens in the S phase. We're making an extra copy. We're going from a single chromatid to a doubled chromatid, right? So that by the time you get here, the chromosomes that did look like this, now they look like this, and they stay like that. And then what happens during mitosis? They get pulled into singletons again, don't they? All right. So knowing that, right? Knowing that, and knowing that the cell is moving through these different phases, which of these phases in the pie chart do you think stage 1 is? Most likely this is G1, right? Where there's only a single chromosome, nothing's been doubled yet, right? Okay. Then stage 2 has got to be the S phase, doesn't it? Because you double the amount of DNA in the S phase, because you're getting ready to send it to the 2 new cells when mitosis happens, okay? Stage 3 is going to be G2, where it's waiting and preparing for mitosis. And then here, right there, is when mitosis is happening, because now you're cutting that DNA back to what the original amount of DNA was, right? Okay. All right, so now knowing that, based on figure 1, which of the following statements correctly links a stage of the cell cycle with the event occurring at that stage? You just need to pick the statement that matches what you just decided. So, A, stage 1 represents the G2 phase? No. It definitely represents the G1 stage. We know that's not right. It couldn't be G2 because G2, you have double DNA, and this says 1 time. Okay. B, synthesis of sufficient DNA for the 2 daughter cells occurs in stage 2. That's a possibility. Let's notate that because it does look as if the S phase, where DNA replication happens, matches what it says in choice B. Okay. C, stage 3 includes mitosis? No, it does not. It only includes stage G2. Okay. The replication of genetic material occurs in stage 4? No. The division of that genetic material back into 2 new cells is happening there. Now here's a point of confusion that I've noticed with a lot of people. You're getting confused between DNA being divided into two sections, which happens during mitosis, and DNA being replicated, which what happens in phase S, in the S phase, right, during DNA replication. When DNA is being replicated, the cell is spending quite a lot of time going through and making an extra copy of the entire DNA strands in whatever cell it's talking about. That's a lot of work. It takes a long time. That's why the S phase is quite time consuming. And it happens way before mitosis, because a lot of work has to be done not only to make all the new DNA so that everything's doubled, but also to make sure that it's correct, that there weren't any mistakes in it, right? To check for errors. Okay. So replicating the DNA, going from a single chromosome to two sister chromatids that can then go into the M phase, that is different than separating those chromatids during mitosis, right? Okay. So DNA replication, you're increasing the amount of DNA, making copies during the S phase. DNA separation or chromosome separation is happening during the M phase, right? Where you're separating the DNA you already made into two new cells. All right, let's go to the next question. All right. Researchers have discovered details about apoptosis. I love the word apoptosis. It's such a fun word. It just means programmed cell death, literally. And that might sound like a bad thing, except think about the fact that you have fingers that can move independently. The only reason you have fingers that can move independently is because at some point in your development, when you were a fetus, they were paddles. And the cells between your fingers were programmed to die so that you had separate fingers. That is actually apoptosis. So apoptosis definitely has its uses, both in fetal development and in protecting you from cells that have gone rogue. So another reason why we would have apoptosis, programmed cell death, is if something wrong is detected in a cell. There's a mistake in the DNA. There's a mutation there that might be dangerous. If a cell detects that and it can't correct the mutation, it will initiate apoptosis so that that cell dies, which is a good thing because that cell might have become cancerous. So that our body does that for us all the time, detecting cells that are problematic, have mutations in them. Oh, not a good cell. Let's get rid of that one. You're still okay. You have trillions of cells in you. There's plenty of cells to take their place. But you don't want cells that are going to be bad. Okay, so back to this question. Apoptosis is being studied by studying embryological development of the nematode worm. Apoptosis is a normal developmental process in C. elegans, which is a scientific name for a nematode. They found several genes involved in apoptosis, including SED9 and SED3. Now here's where I actually made notations for myself. Okay, SED9 and SED3. These are genes. Remember, genes are sections of DNA that are responsible for making certain proteins, okay? Now, when I did this, I then made sure I knew what each gene was supposed to do. So it says, the SED3 gene was found to promote cell death. So the SED3 gene says, die to the cell. Yes, go do it, die, okay? And the SED9 gene serves, and then the SED9 gene inhibits it. So it says don't die. Okay? The SED9 gene serves as a regulator that prevents apoptosis in the absence of a signal promoting apoptosis. Which of the following statements best justifies the claim that changes in the expression of this, meaning if you mess with this or mutate it, what happens? Okay? All right. So choice A says an experiment showed that a mutation, right, in the SED9 gene, if you mutated it, led to excessive death in C. elegans. Does that make sense?